Hello, everyone. Today, the topic we would like to present is microbial upscaling and degradation of plastics. As we can see in this figure, plastic ends up in the soil and oceans, which is essential homes for living organisms. According to a study report that is published in 2012, approximately 165 million tons of plastic were present in the world's ocean. The persistence of plastic pollutants in the water body is a major concern because it causes serious hazard to organisms' health. It is well known that organisms, for example, plankton, fish, and human race, ingest these high carcinogens through the food chain. The consumption of fish that contain these toxic substances can also cause cancer, various immune disorders, and several congenital disabilities. A data generated in 2015 by Economic India Times showed a production of 6,300 million metric tons plastic over the world. And out of total, 79 is accumulated in landfills, ocean, and other water bodies, while 12% is incinerated, and only remaining 9% have been recycled. If we don't take any actions, roughly it is estimated that 12,000 million metric tons of plastic waste will be in landfills by 2050. We actually have already discovered a few options to deal with the plastic waste. We can simply recycle it or upcycling it to value-added products. Also, we can degrade it. Today, we want to focus on microbial degradation. And first of all, some basic concept that we need to get familiar with. The first one is that several steps are involved in the microbial degradation of plastic polymers, such as biodeterioration, biofragmentation, assimilation, and mineralization. And also the process of biological deterioration of plastic pollutant depends on a lot of factors, such as surface area, functional groups, molecular weight, hydrophilic and hydrophobicity, melting temperature, chemical structure, and crystallinity. Microorganisms as a popular option for plastic degradation are perfectly suitable for the task of reduction of plastic contaminant because they acquire enzymes that permit them to utilize plastic as a substrate. Microorganisms can catalyze the oxidation reduction reaction to break down the chemical bonds in the plastic polymers.
The exponential rise in plastic waste production is resulting in an environmental crisis that demands cutting-edge technologies for recycling post-consumer plastics. Enzyme-mediated biocatalytic depolymerization has surfaced as an effective and eco-friendly solution for plastic processing and recycling. Numerous plastic degradation enzymes have been identified from microbial origins. Currently, protein engineering technologies are being utilized to modify and optimize these plastic degrading enzymes. Utilizing protein engineering techniques can enhance the catalytic performance of plastic degrading enzymes is an emerging area of research. Generally, protein engineering encompasses two approaches. The first one is rational design, and the second one is directed evolution. Rational design involves modifying the protein based on its structure, mechanistic properties, computational simulations, and modeling. Most current studies on engineering plastic degrading enzymes rely on rational design due to the available structural and mechanistic information for many such enzymes. The absence of efficient high-throughput screening methods poses a significant obstacle for the directed evolution of plastic degrading enzymes. The only known attempt to use directed evolution for engineering, for example, PHP depolymerize, did not yield any variants with improved activity. So instead, we will concentrate on discussing rational design strategies for enhancing plastic degrading enzymes. The first approach we're going to introduce is enhancing enzyme thermal stability, particularly for plastics with high glass transition temperatures. However, the low thermal stability of naturally occurring plastic degrading enzymes is a major bottleneck for practical applications. Several strategies have been proposed to improve enzyme thermal stability, including the first one is to introduce disulfide bonds or salt bridges, which are essential for protein folding and thermal resistance. The second one is the introduction of hydrogen bonds. It involves engineering the formation of hydrogen bonds in regions responsible for more stable enzyme structure, helping to help maintain higher order structures and to improve its resistance to high temperatures. The third one is introducing more protein residues to stabilize the protein tertiary structure against high temperatures. The last one involves introducing glycosylation in eukaryotic microbial cells to enhance enzyme thermal stability through thermodynamic stabilization and prevention of thermal protein aggregations. However, it is important to note that enhancing thermal stability can occasionally impair catalytic efficiency due to interference with the active site. In most cases, engineering enzymes with improved thermal stability also exhibit improved plastic degradation efficiency. For example, goodness from AHK 119 has an increased melting temperature from 74 degree to 79 degree. And goodness from another AHK, AHK 190 also has an increased melting temperature by 3.7 degree. The second approach is called reinforcing the binding of substrate to enzyme active sites. So the active site region is crucial for engineering plastic degrading enzymes, as it plays a key role in determining the efficiency of plastic depolymerization. A common strategy involves creating a wider opening of the active site to increase plastic substrate accessibility. Successful examples including modifying enzymes like Fusarium solani goodness, PETase to enhance degradation activity towards PET and PBSA plastics. However, a larger substrate binding size does not always guarantee improved catalytic performance, as it may lead to weaker substrate affinity. In some cases, a narrower active site may be more effective, as seen with PETase when modified with double mutation S238F and W159H, leading to more efficient degradation of PET and PEF. The hydrophobicity of the substrate binding group is another potential target for engineering. Increasing the hydrophobicity can enhance degradation efficiency due to higher affinity. Simultaneously tuning the hydrophobicity and opening size of the active site can synergistically improve enzyme performance, as demonstrated with a T-Fusca cuteness mutant. In summary, it is important to note that for both approaches, it is essential to carefully balance these changes to avoid reduced substrate affinity or weaker binding ability. One example for this approach is by enlarging the opening size of active site cleft, PET hydrolase has an improved PET degradation activity, as well as the capability of hydrolyzing catalyzing PET from commercial bottles.
The third approach is improving the interaction of the substrate and enzyme surface. The substrate binding process is detected by the electrostatic and the hydrophobic interaction between the substrate molecules and the amino acid residue on the enzyme surface. Therefore, modification of surface electrostatic and hydrophobic properties are kind of strategies, making the enzyme surface electrically neutral and might reduce electrostatic repulsion between the enzyme and the plastic substrate, thus enhance binding the degradation efficiency. On the other hand, turning the enzyme surface with increased hydrophobicity could also enhance enzymes and substrate interaction to increase the surface hydrophobicity of enzyme, such as through surface modification or immobilization on hydrophobic surface. The hydrophobic environment around the active site can be enhanced. This can increase the strength of hydrophobic interaction between the enzyme and the substrate, leading to improved binding and catalytic efficiency. Another approach to enhance substrate and enzyme interaction is fusion of binding accessory to enzyme surface. This method is inspired by the fact that some enzymes evolve in nature. Biopolymers degradation process and accessory binding domain specialized in polymer substrate adhesion. Therefore, plastic degrading enzymes that lack this function can be fused with heterogeneous binding modules to enhance their interaction with plastic surface. Furthermore, for refining enzyme functionalities, can fall in three types of enzyme engineering. Plastic degradation intermediates or products can inhibit enzyme activity to mitigate this, modifying the active architecture has been shown to be effective. For example, mutation G62A in the substrate binding groove of the TF-Cat2 results in 5.5 times decreasing in the binding constant for inhibitory hydrolysis products, thus leading to improved PET degradation. Additionally, fusion with the ancillary enzyme capable of the decomposition of the intermediate compounds has also been demonstrated to be effective in reducing products inhibition. Also, enabling catalytic promiscuity is another approach to expand the capacity of plastic degradation enzyme to use different plastic substrates, especially for plastic with few enzymes known to efficiently degrade them. For example, by manipulating the Cat1 casting, researchers were able to develop its polymerase amylase activity towards the depolymerization artificial PAS, resulting in a 6 to 15-fold higher hydrolytic activity. Finally, fusion of plastic degradation enzyme with another auxiliary enzyme has been exploited to create a functional catalyst for improved polymerization efficiency. By constructing a lipase for cutting fusion protein, researchers were able to achieve remarkable higher PCL hydrolysis activity, while synergistic fusion of PETase and MHETase can improve PET hydrolysis efficiency by threefold when compared with widely type of PETase. So here is all we know about the protein engineering for rational design strategies for improving plastic degrading enzymes. If you want to see more information of above methods, you can go to the reference page to find more information. So thank you all.